A nucleotide is the building block of DNA. It has three parts: sugar, phosphate, and a base. They dynamically link together to form the genetic code.
Every nucleotide is built from three basic components. The phosphate group is key, composed of phosphorus and oxygen. It locks these molecules together. This creates the sturdy backbone of DNA and RNA. DNA contains a pentose sugar. This 5-carbon sugar is called deoxyribose. It lacks an oxygen atom. This occurs at the 2' carbon.
Nitrogen bases are the building blocks of DNA. There are 4: adenine, thymine, cytosine, and guanine. They pair up specifically to store genetic information.
Purines are key components of DNA and RNA. They feature a standard 9-atom numbering system. Adenine and guanine are the primary purines. Both share this fused double-ring structure.
Adenine is one of the vital purine bases. It features a fused double-ring structure, a pyrimidine ring joined with an imidazole ring. This structure is key to encoding genetic information.
Guanine is one of the 5 primary nucleobases. It is classified as a purine derivative, featuring a fused double-ring structure with a distinctive carbonyl group.
Adenine has an amino group at position 6, an oxygen 6, and an amino at position 2. Adenine pairs with cytosine.
Pyrimidines are single-ring structures. They include cytosine, thymine, and uracil. Uracil is found in RNA. They pair with purines.
Cytosine is a pyrimidine base. It features a single 6-membered ring structure, with an amine group at position 4 and a ketone group at position 2. To form a nucleotide, a pentose sugar acts as the central base. A phosphate group attaches to 5th carbon, forming a strong covalent bond. Finally, a nitrogenous base joins.
They join as a phosphate connects to a sugar. This forms a strong sugar-phosphate backbone, creating a single strand of DNA. DNA is formed by pairing specific bases. Purines must always join with pyrimidines. Adenines connects perfectly to thymine, while guanine binds only with cytosine. This creates the iconic double helix.
The DNA molecule is shaped like a double helix. 2 long strands twist together like a spiral staircase.
The strands are anti-parallel. One strand runs in a 5' to 3' direction, while the other runs 3' to 5'. The outer sides consist of alternating sugar and phosphate groups. This forms the structural rails of the genetic ladder. The diameter of the helix is 20 angstroms, or 2 nanometers.
Did you know that in DNA, each adjacent base pair is stacked exactly 3.4 angstroms apart? That's a tiny 0.34 nanometers.
Single complete turn of the DNA double helix measures exactly 34 angstroms, and within this decimal spectral length, it contains about 10 base pairs.
Ever wonder why DNA is a helix? It comes down to the bonds between the phosphate, sugar, and base. These bonds force an attachment at 120 degrees instead of a flat 180 degrees, creating the twist.
The larger groove is called the major groove and occurs when the backbones are far apart, while the smaller one is called the minor groove and occurs when they are close together. The DNA double helix is a marvel of structure. Hydrogen bonds connect the base pairs in the center, while hydrophobic interactions stack them safely inside. The sugar-phosphate backbone of DNA is negatively charged. This makes it hydrophilic, allowing it to bond with water.
To form a nucleosome, specific proteins are necessary. These are histones H2A, HB, H3, and H4. Finally, H1 locks the structure in place.